backbone of this tree, where you have the M269, and then a clade underneath it defined by P311, and then if we have time we'll get into some of these sub-branches beyond that. So just taking that simple tree and color-coding the pie charts, this is what the pattern looks like across Europe. Notice that the white areas indicate no R1b at all, and as you move towards the west you have upwards of over 80% of the Irishmen having this R1b lineage. And then the different subgroups within R1b are color-coded. We're going to try and make sense of this pattern. So squint at it, see if you can see any patterns in there. Maybe you'll come to your own conclusions. But I'm going to give you some ideas. Now, as we just said, we have this incredibly high frequency of R1b in the far western parts of Europe, and decreasing amounts of R1b over as you move towards Eastern Europe and the Middle East. Now, because of this very, very high prevalence of R1b in the British Isles, in Western Europe, 50% in Central Europe, it was assumed that these must be very old lineages, must have been here since the Paleolithic, since humans first got into Europe. That would explain their very high frequency. But recent studies are overturning this idea and suggest something much more recent for the appearance of R1b in Western Europe. Now let's talk about the different ways that R1b could have arrived in Europe. First, as I just suggested, is that it may have come in with the first anatomically modern humans. And if you saw Chris Stringer's talk yesterday, he told us that modern humans appeared in Europe around 45,000 years ago. So, and making it all the way into England as recently, or as old as 43,000 or 41,000 years ago. So it could be that these initial migrants from Africa carried R1b, and that's why we see what we see today. Then about 24,000 years ago, as also Chris mentioned, Northern Europe was under an ice cap practically, and people basically died out and retreated to the south into these refugia in Southern Europe. And then when the climate warmed up again about 4,000 years later, about 18, 19, 20,000 years ago, there was an expansion out of these refugia from Spain, from the Balkans, back into Europe. So it could be that the patterns we see today reflect this last glacial maximum reinvasion of Europe. And finally, we also know that the Neolithic made a huge impact on Europe. Farming got started in the Near East around 10,000 years ago, and farmers outpaced reproductive abilities of hunter-gatherers and replaced them, at least it's thought to some degree replaced hunter-gatherers in many parts of the world, including Europe. So it could be that the Neolithic is the time that we should be paying attention to explain the existing patterns of Europe. Now, just a moment about sort of the background, the scholarly background about the Neolithic transition. There are thought to be at least 9 different centers of origin of agriculture independently in different parts of the world. And Jared Diamond and Bellwood in 2003 put forward an interesting hypothesis that during the Neolithic transition, everywhere in the world, the genes of the food producers replaced to some degree or completely the genes of hunter-gatherers. And that left huge impressions on the patterns of genetic variation that we see today. So with that background, some of the researchers working on the Y chromosome began to explore the hypothesis that the Neolithic was the major transition period for genetic variation in Europe. So the controversy breaks down to, we know that the relative contributions to Europeans of both hunter-gatherers and Neolithic farmers is in question, has been debated intensely for a number of years. The hypothesis was that M269 or haplogroup R1b was Paleolithic, started to degrade but we know that it's carried the most frequently in Europe by over 110 million men. So recent studies to start sort of to erode away this Paleolithic hypothesis and agree that now that the Neolithic was the important point of turnover, but they disagree exactly as to when in the Neolithic. So let's take a look at these two ideas. The first came out in 2010, a paper by Mark Jobling's lab at Leicester, first author Ballares, where they examined, you can see here on the left, they have the dates of the early Neolithic sites into Europe, the frequency of the R1b again, And then they looked at the microsatellite diversity on the background of R1b. So you can look at any number of STR markers on the Y chromosomes of these men and ask, where do you find the most diversity? And under the center of origin hypothesis, where you find the most diversity points to the origin where things began, and you look for gradients of decreasing diversity from there. In fact, they showed that the microsatellite diversity was the greatest on R1b in the Near East, and it decreased as we moved northwest of Europe. The timing of that, they estimated, put it squarely in the middle of the Neolithic, and so this spreading of diversity from the Near East clearly must have been with a Neolithic transition. So they concluded that the chromosomes of Metallist gatherers nearly replaced those of incoming farmers, were nearly replaced by incoming farmers, and there was a link between this technological advantage the farmers had over the hunter-gatherers that led to the replacement of the hunter-gatherer Y chromosomes. In contrast, most of the maternal lineages descended from hunter-gatherers, suggesting reproductive advantage of farming males over hunter-gatherer males. So, by looking at the mitochondrial DNA story, it didn't look quite the same. There was persistence of Paleolithic mitochondrial lineages. Therefore, it looked to be more driven by men, this turnover, especially by looking at the Y chromosome or looking at the turnover in men. Then just a couple of years after that, a cautionary tale told by Busby et al. They looked at the same data that Ballarès looked at. They looked at an extended amount of data. They redid the analysis and didn't find quite the same results. They did not find a clear trend in the decrease of diversity as one moved from the Near East to the Northwest, which was the major tenet of the hypothesis that it was the Neolithic transition itself that led to the spreading of R1b into Europe. They were not able to get reliable estimates of the TMRCA or the ages of these Y chromosome lineages. So basically, rather than telling us what happened, they told us what didn't happen. They said it was unlikely that it was spread during the Neolithic. But they put forward a couple of interesting ideas. They looked at, again, this M269 grouping with the P311 subgrouping. When you look at the entire clade, it looks as what we've shown. If you look just at the R269, M269, you can see that these lineages are in fact restricted to the Near East, and therefore, when they looked at P311, it showed this pattern again, suggesting, yes, there was an older center in the Near East and a recent spreading into Western Europe, but the timing of that was not Neolithic so much as it was post-Neolithic. So, the two hypotheses from these different groups, A311 originated prior to the Neolithic and spread into the Western Europe, which would predict that diversity gradient, which is not found. Or P11 originated more recently, and its current distribution is the result of major population movements after the Neolithic. So we've gone from the Paleolithic hypothesis to the Neolithic hypothesis to now something post-Neolithic. They didn't have evidence to support either one of these strongly. So now I want to turn from the indirect approach of studying contemporary variation and actually say we have enough data now to look at what was in the ground at the time of the Neolithic to see which Y chromosomes were there. This is a direct test of these hypotheses. So we now know from a few different studies, one in Germany with 3 different Y chromosomes discovered at about 7,000 years ago, the LBK culture. A larger sample in southern France, about 22 Y chromosomes have been typed there. Their age is about 5,000 years ago, near the end of the Neolithic. And then we have these 2 chromosomes in northern Spain that were typed from this cave in, at 5,000 years ago at the end of the Neolithic. So both of these about 5,000 years ago, this about 7,000 years ago, and this is the famous sample from the Tyrolean Iceman, Ötzi, who is dated to about 5.3 thousand years ago. So we have all of these different Y chromosomes. The question is, do we find R1b here during the Neolithic supporting the hypothesis of a Neolithic entrance? When we actually look at the results, you can see that in the German sample there was 2 G chromosomes and 1 F star, no R1b. In France there were 20 G2As, 1 I2, no R1b. Spain, we had 5 G2As and 1 E, a very frequent E type in Europe from Africa. And Utsi himself is also G2A. So the striking thing is, A, there's no R1b present in these samples from the Neolithic, and B, we have a high frequency of another Y chromosome type, G2A. Now, we don't usually need statistics when the numbers are that overwhelming, but if we do apply some very simple Fisher exact tests, we can look at contemporary samples from southern France as close as we can find to the ancient DNA sample, and we can ask, the R1b frequency of these is 60%, 52%, 53%, and it's 0 here. It's a very simple way of asking if 0 out of 22 are R1b versus 94 in these samples out of 172. It's a simple test. The p-value is highly significant. These are not being sampled from the same population. If we look at the entire data set across Europe that we just described, you have 0 R1b out of 32 ancient samples, whereas you have 1,400 out of a total of 2,410, which obviously is also very significantly different. Just to make a point about this G2A, that seems to be the signature of the Neolithic, not R1b. This is the current distribution of G2A, and here's the spread of agriculture into Europe. It seems to be also a nice overlay. So I guess we can imagine that these Neolithic men carried a great proportion of G2A Y chromosomes, which today are very, very infrequent in Europe as they were replaced by something else, namely R1b. Now, what about the mitochondrial record? This is something that was taken from a study by Brandt et al. Recently. And what they've done here is they have the timeline here going from left to right to present. And then they have something in the Neolithic time period. And they summarize various mitochondrial lineages into 3 different groupings: the gray, the brown, and the yellow. The gray being the hunter-gatherer mitochondrial types, the U mitochondrial lineages, the brown being the early Neolithic lineages, brown, and the yellow being the late Neolithic. And the point I want to make, and then a bunch of other chromosomes here in the dotted line, even though there are major changes in the frequencies, and this isn't quite fair because these haplogroups are grouped into these groupings, we're not looking at them individually, you can see that the pre-Neolithic or early Neolithic has a bunch of hunter-gatherer Y chromosomes, and they drop off, but they don't go extinct. They survive to present. And then the Neolithic Y chromosomes maintain some intermediate frequency throughout the Neolithic into the present being over here. So the point I'm trying to make here is that these mitochondrial lineages are continuous throughout the Neolithic and do not get entirely replaced by whatever process replaces the Y chromosome lineages. So there's a difference between the Y and the mito. I tried to summarize this in terms of pie charts. If we look at the Y and the mito here, and the late Neolithic and the contemporary pop frequencies, here's what I showed you from the ancient DNA data: a great preponderance of G2A, today a great preponderance of R1b, and here are the other minor frequencies of these lineages, which today, some of them still exist. The F star seems to disappear, and we also have the addition of a couple of other lineages. I is probably a very ancient lineage that has survived through to present day, through hunter-gatherers. However, you can see, even though there are different frequencies between the late Neolithic and the present, we still see the same basic pattern for the mitochondrial DNA. So, to summarize all of that, the combined evidence supports the hypothesis that the current distribution of the P311 lineage is the result of the major population movements occurring after the Neolithic, and the populations carrying the P311 chromosomes appear to have nearly displaced Western European Neolithic Y chromosomes, which is quite different than what we see in the mitochondrial side. So what conferred this advantage to these post-Neolithic men, allowing them to replace the Neolithic farmers' Y chromosomes? And I would argue that this Neolithic turnover occurred during the Metal Ages, and it's the metal itself that conferred the advantage, possibly through weaponry. So here we have the Neolithic, the Bronze Age, the Iron Age, and the Bronze Age is actually the first time humans used metal to specifically create weapons. The sword, the spear, and the shield were all invented in this period, and the warrior identity became a standard part of daily life as represented by cultural record. The Iron Age, which followed the Bronze Age, the first true mass production of weaponry, because iron was so prevalent throughout Western Europe, whereas tin and copper were quite rare. This revolutionized not only weaponry, but it revolutionized agriculture. And then we also know between 1200 and 1000 BCE, the Celts were the most dominant Iron Age warriors and raged across much of Europe. So we can imagine that there was something going on between Celtic warriors or other warriors on chariots, and chariots were also an invention of this era. Now all of this stuff seems to have spread with Indo-European languages. So not only do we see the spread of Y chromosomes coming from the steppes into Western Europe, we see the spread of Indo-European languages. This shows the spread of the chariot. And here's a coin from, I believe from the UK, an old coin that shows a good example of the introduction of the horse. There are domesticated animals, other such as sheep and cows that came in also. So we have a bunch of changes in the technology and the culture summarized by metal weapons, chariots, horses, domesticated animals, less reliance on agriculture, a stronger reliance on herding and the drinking of milk. So all of this was happening post-Neolithic, and so it seems to be a good hint as to what might have been causing this big turnover in Y chromosome variation. So just to summarize, in the early Neolithic, R1b was probably restricted to the Near East, didn't start spreading until the late Neolithic, but still very restrictive into this part of Europe. If you look at the red, the Mycop and the Yamna cultures were places where these metal workers were active at the time, but it had not spread at this point. Then during the Middle Bronze Age, this likely is the time at which R1b was able to penetrate into Europe, into Central Europe first, and then into Western Europe later. And then late Bronze Age, I would like to say that once the spread of R1b came in, there was differentiation within R1b that's quite interesting, and I'll turn to that point next. And the renewed expansion could have continued as recently as the Hallstatt and La Tène cultures very recently, perhaps with Celtic warfare. Now, once R1b got into Western Europe, it's really clear that there were different centers of expansion locally that continued the spread of subclades of R1b. So you have U106 at 44% Friesland, you've got L21 at 70% Ireland, you've got U152 in the Alps. So the radial distributions from each of these centers is also very interesting and brings us even closer to the present. And it's very interesting that the pattern suggests differentiation across Europe via multiple geographic centers of expansion. So we now have a new hypothesis, a post-Neolithic center of renewed expansion that is very consistent with the archaeological record, which shows that there were many different centers of archaeological expansion that correlate quite nicely with these different Y chromosome subclades. So I've put together this cartoon of what could have been the case. You have M269 originating in the Near East, the P11 mutation subsequently originating and spreading into Europe, and then once in Europe there were these different centers of re-expansion, with this lineage going into the Iberian area, L21 being the major lineage in the UK, and U106 going north, and U152 going south from the Alps. Now let's just focus for a moment on the L21 expansion into the UK. I would just like to give you an idea of the kind of variation we're seeing on the SNPs on the background of this particular lineage, which explains most of the lineage variation in males in the UK. There are now a number of SNPs that are highly informative, that are starting to show very interesting differentiation across the UK, the British Islands. In Ireland, you've got a high frequency of this DF23 lineage,
You've got, in Scotland, you've got a very high frequency of the CTS 11722 SNP, and in England, notice DF21 is quite frequent relative to the other parts of the UK. And I think this kind of thing, information where we have all these new SNPs and many testing for these new SNPs should give us a great deal of information about the variation within these regions and how it differentiated and went across the British Isles. So that could be a reasonable place to stop. I did want to say that the story becomes even more perplexing at some levels when we take a bigger step back. We have a paper about to be published where we're revising the structure of haplogroup K. So this is the entire Y chromosome tree. Here's the lineage R that I was just talking about. R is part of a bigger group called K. So up until recently, K was just kind of this nondescript group with many groups coming off one basal branch. With the new SNP information we're gaining, we can see that the K group now is split up into subclades that weren't visible before. The R lineage is part of the P and Q group. Q is the most frequent lineage in the Americas. And now we can see from this structure that the nearest neighbors to R, P1 and P star, will represent the ancestral forms of R that are of interest to us. It's interesting that the closest relatives to R, the P lineages, are now found to be restricted entirely to Southeast Asia. So the ancestor of R, before it got to the Near East and spread from there, must have come from somewhere further east. When we look at the frequency of this P star group, which is the ancestor, again, you can see it's this P295 star, you can see it's restricted to these places, Southeast Asia and Western Island Southeast Asia. So what we were able to determine was there was a rapid diversification of the entire K subclade in Southeast Asia, giving rise to P27, which led to this group, and a later appearance of R and Q in Western Eurasia. Just to summarize all of that, it seems that you have the out of Africa events. There's a period in Southeast Asia where things rapidly diversified, and at some point, the ancestors of R made it back to the Near East, and then expanded with farming, with the Neolithic, with Indo-European, in the various directions that we see. There was even an incursion into Africa of R. So, I guess we're talking about some things that are not well accepted, but seem to be pointing in the direction of an unusual pattern for the origin of the entire R group, both its ancestor in Southeast Asia, and then the very, very recent returning over of Y chromosomes in Western Europe associated with the Metal Ages. And so I'd like to stop there and open it up for any questions that you might have, acknowledging the various people who contributed to this work, both at Family Tree DNA at my lab in Arizona.